OxyMBBS. One-step solution for all medical studies.
Now friends, let's talk about the bone remodeling. So it is a process in which old or injured bone tissue is replaced by new or healthy bone tissue. As this is a bone and this is the cross section of the bone taken like this, and let's suppose this part of the bone is either old or damaged. So this part of the bone needs to be replaced by new or healthy bone tissue. Now friends, I am showing the enlarged view of this part here for better understanding. As you all know, bone, either it is old or new, has cross-linked osteocytes. So these are osteocytes, and these blue lines are collagen fibers, and in between collagen fibers, these are what? These are crystals of hydroxyl apatite. Okay, it means friends, these hydroxyl apatite, collagen fibers and osteocytes are now become old. That is why they needs to be replaced by new bone tissue. As you all know, old things are very weak. That is why if they will be present in the body for longer duration, then they may cause fracture of the bone. So friends, to prevent this fracture, the old bone tissue must be replaced by this new bone tissue. Now friends, let's see how this whole process of remodeling takes place in the bone. As you all know friends, the osteocytes play a very important role of monitoring. Okay, so these osteocytes sense that the bone tissue which is surrounding these osteocytes has become old. That is why it needs to be replaced by new bone tissue. Okay, so for it, these osteocytes give signals to the osteoprogenitor cells. These are osteoprogenitor cells which are present in the periosteal inner layer of the periosteum and endosteal layer to produce more and more osteoblast cells, and these are what? Osteoblast cells. And now the functions of these osteoblast cells starts, and the osteoblast cells secretes roots proteins. So roots are the proteins. So from this R, it is rank ligand protein, and for this rank ligand we use rank L as short form. From O, it is osteoprotegerine protein, and for this OPG is used. And from this O, T and S, these are osteopontin, sialoprotein and thrombospondin proteins. Now friends, we will see what are the functions of all these proteins. Now as you all know, this giant multinucleated cell having a ruffled border is which cell? This is osteoclast cell, and this osteoclast cell is formed from approximately 2 to 50 monocytes and macrophages. But my dear friends, this fusion is not spontaneous. This fusion depends on a protein, and that protein is called rank ligand. So for the attachment of these monocytes with these rank ligand, these monocytes must have a receptor for these rank ligand, and that receptor is called rank. So these monocytes without a rank ligand are called undifferentiated or inactive monocyte. But after attachment of this rank ligand to this receptor, there are some changes occurs in these monocytes, and these monocytes are now differentiated or they are active monocyte. And friends, only and only these differentiated or active monocytes are now able to fuse with each other to form a giant cell that is called osteoclast cell. So these are newly formed osteoclast cell, and these cells reaches at a place where bone degradation is needed. As bone degradation is a long process, that is why these osteoclast cells must be firmly attached with the surfaces of the old bone, and this function of firmly attachment is done by these proteins osteopontin, sialoprotein and thrombospondin, which firmly attach osteoclast with the extracellular matrix. And after firmly attachment, these osteoclast cells start to secrete bone degrading substances or agents through these ruffled border. Okay, and the breakdown agents like protons which creates acidic environment which catalyzes the inorganic salts of calcium and phosphorus, and collagenases plus cathepsin K causes breakdown of organic matrix like collagen fibers. So these are pieces of old bone having organic and inorganic material, and these breakdown pieces are reused by the body. So for it, these breakdown pieces are taken up by the osteoclast cells through their ruffled border by the process of endocytosis, and by the process of exocytosis through the cell surface opposite to the ruffled border is sent into the blood, and these collagen proteins, calcium, phosphorus and many more nutrient substances are again reused by the body. As this part of the bone has broken completely, now it's the time to break this part, and for it these osteoclast cells moves forward, and for new bone synthesis at this place, now osteoblast cells will come at this place, and for the synthesis of new bone at this place, these osteoblast cells are now ready, and this is the newly formed bone. And similar to this, the remaining part of the old bone is first degraded, then new bone is synthesized at the degraded places. Now again, what is bone remodeling? From in the remodeling, it is a resorption of bone, and from modeling it is bone formation. So here is bone formation. So remodeling is actually combination of bone resorption and bone formation. And if there is only bone formation without bone resorption, then it is called only modeling. So up till now, in the process of bone remodeling, we have seen the functions of these rank ligand and these proteins, and now we will discuss the importance of these osteoprotegerine protein. So with the help of these osteoclast cells, this old part of the bone has degraded, and in place of it, new bone is synthesized. Now to prevent the further activity of these osteoclast cells, as if their activity will not be stopped right now, then they may cause degradation of normal tissue also. So to prevent the further activity of these osteoclast cells, a protein that is called osteoprotegerine is needed, as these osteoprotegerine proteins binds with the rank ligand and prevent the attachment of rank ligand with the rank which is present on the undifferentiated or inactive monocytes. And because of this, these undifferentiated or inactive monocytes will now not be able to convert into the differentiated or active monocytes, and because of this, osteoclast cells will not be produced. And these osteoprotegerine also decreases the activity of persistent osteoclast cells. In this way, friends, because of these osteoprotegerine proteins, production of osteoclast cells as well as activity of the osteoclast cells is decreased. Now, how will you remember that this osteoprotegerine is related to prevention of rank ligand activity? It is very simple, friends, as this PR in the protegerine will remind you that it is related to prevention. It is related to prevention, and prevention of this rene in the osteoprotegerine. Rene means rank ligand. So osteoprotegerine prevents activity of rank ligand. Now let's talk about the factors which are affecting the bone remodeling. So first one is exercise. If somebody is doing heavy exercise like running or weightlifting, then it may cause stretch or stress on these processes of the osteocyte cells. And because of this persistent stretch or stress, these osteocyte cells give signals to the osteoblast cells, and osteoblast cells start to synthesize new bone. And as you all know, in the adult, that is at around 20 to 25 years of age, the bone cannot increase in the length, but because of this extra deposition of bone tissue, bone increases in the width, and this is a bone after increase in thickness. And now this thick bone is able to bear heavy exercise. So during heavy exercise, only bone formation takes place. That is, only modeling takes place in the bone. And opposite of exercise is rest. If somebody is doing rest, then osteoclastic activity increases, and because of this, thickness of bone decreases. So this is a bone in a person who is bed resting for a long duration. So during rest condition, only resorption activity takes place in the bone. And if somebody is taking vitamin A in a high amount, then it causes increase in the thickness of the bone. Now friends, let's talk about how parathormone and calcitriol, that is also called vitamin D3, regulates calcium level in the blood. This is osteoblast cell, and these are receptors for the parathormone, and these are receptors for the vitamin D3. When in the blood calcium level falls, then either vitamin D3 or parathormone binds to their receptors on the osteoblast cells, and after binding they give signals to the osteoblast cells to produce more rank ligand proteins and to decrease the synthesis of these osteoprotegerine proteins. As you all know, these osteoprotegerine proteins prevents the activity of rank ligand. That is why these osteoprotegerine proteins are also called decoy agent for the rank ligand. And now, as you all know, what these rank ligand will do. So here is surface rank ligand protein, as these are undifferentiated or inactive monocytes, and this is rank, that is a receptor for the rank ligand. So the cell surface rank ligand binds with the cell surface rank and activates monocyte, and the monocyte is now differentiated or active. As in this differentiation, first one cell contacts with the other cell. So it is cell to cell contact differentiation. And now, as you all know, what these differentiated or active monocytes will do. They will form which cell? Osteoclast cell. And as you know, osteoclast will do what? It will cause degradation of bone tissue. And in the bone, what is present? Calcium, phosphorus and collagen protein. And this calcium from the degraded bone is sent into the bloodstream, and in this way level of calcium increases in the blood. So this is how calcium is regulated in the blood by these two hormones.